This PowerPoint is the second Microevolution PowerPoint, therefore it is also the last one before your test. We're going to split it up into 2 to 3 videos. You only need to watch the first video for tomorrow, which is Thursday. The date would be the 22nd. When we talk about microevolution, we are focusing specifically on genetics and alleles. We are not looking at big picture evolution. Evolution is defined as a change in inherited characteristics of biological populations over successive generations. But to get to those big picture changes, we need to look at the generation to generation changes. So every time we reproduce, which is microevolution, looking at the allele frequencies or the different genetics that pop up within a population over time. The change in the genetic makeup of a whole population is impacted by 4 processes: mutations, selection, which is both natural and artificial, gene flow, and gene drift. Make sure you know these 4. To determine the allele frequency of a whole population, let's look at our very simple frog population. On the left, the color is determined by a single gene, which has 2 alleles or 2 options, and when you have the 2 different alleles, it's going to exhibit as incomplete dominance, or a totally different color, right? We know if one is totally dominant, it will always mask the other. This is not the case with our colored frogs. If the frog has 2 G alleles, it will be green. If it has 2 R alleles, it will be red. And if it has 1 G and 1 R, it's going to be, it says blue, but purple. Keep in mind that if this was a dominant recessive situation, green would always cover up red, or red would always cover up green, but because when we see the 2 present, it's a different color, we call it incomplete dominance. The question is, how do we calculate the allele frequency of the gene pool? And this is a very simple mathematical population. If we look at all of my green frogs, purple frogs, and red frogs, all I do is I go through and I count out each individual allele. So one green frog accounts for 2 CG alleles. If we look, I've calculated up all of the CG and CR alleles. I divide it by the total number of alleles, which in this case is 32, and I get a percentage. We find that the percentage of the CG allele is 75%, CR is 25%. Now let's suppose that the 4 green frogs enter the population. How do the frequencies change? We see that I add more CG alleles, increasing the percentage of that within the population, 80% as opposed to 20% CR. How do the allelic frequencies change if 4 green frogs leave the population? Immigration is coming in, emigration is coming out. We see that there is a decrease in the CG allele because my green frogs left, an increase in the percentage of CR alleles. The impact of these changes on small versus large population. Hopefully we can predict that if we look. If there was 4 frogs joining a small population, we're going to impact the percentages much more greatly than if they joined a larger population. If we look on the right is this is the large population in general, before the frogs join and then after. We see that the percentage only changes 2 to 3 percentage points because there's such a large pool already, which should start us thinking about the idea that the bigger my population, the more stable the genetics are going to be. We see in this small population, my percentages change greatly, or the frequency changes greatly, because I have a smaller group of organisms to begin with. So the first thing that impacts the genetic makeup of a population is genetic drift. Small populations can experience changes in these allelic frequencies much more dramatically than large populations. Also, small populations, genes are also lost. So genes come in and they leave very quickly. When there's only one allele left for a particular gene, then the gene is said to be fixed, thus there is no genetic diversity. Genetic drift, or allelic drift, is essentially the change in the frequency of a gene in a population due to random sampling in the absence of a selective pressure. This is important when populations are made very, very small by any variety of things, hunting, predation, environmental factors. A ton of genes are lost, and negative genes can increase as well. In genetic drift, essentially what happens when there are a few copies of an allele, the effect of this genetic drift or this big change is larger. And so we see that genetic drift is like the loss of a large chunk of genes or the addition of a large chunk of new genes. Genetic drift is really profound in populations that are very small, obviously, because it impacts the percentage of the alleles more dramatically. The most common way that we reduce a size of the population is what we call a bottleneck effect or a bottleneck event, in which the original population is very large, and then something, an environmental catastrophe, cuts down the population. Genetic drift also occurs when a small population arrives at a new habitat or an island, and we have an isolated chunk of the alleles in a new environment. This example is the bottleneck effect. Essentially, we see that there are these prairie chickens. They kind of look like a mix between turkeys and chickens. We see a huge decline in population. When we look at the DNA evidence, we see that the diversity of the genetics has changed drastically. When we had a very large population, the diversity was very large. But that small population means that the birds have lost almost all of their genetic diversity because we killed off and got rid of a lot of the alleles. This is an example of genetic drift. This also means that my population is very unstable. Less than 50% of the eggs that were laid actually hatched. Populations outside of this don't experience this problem, and so the genetic drift and the bottleneck effect resulted in some negative genetic traits being passed on. This genetic drift in smaller populations changes the allele frequencies. A greater change of allele frequencies due to gene flow is evident in really small populations. I've said that a million times, and I think that is the take home, is that smaller population, genetic drift is going to impact it a lot, and the way that we get a smaller population is a bottleneck event. Even if those populations rebound, we see that there is limited diversity because they're still breeding from the same gene pool from the smaller group. The founder effect is going to be another way that we change the allelic makeup of the gene pool. It's the loss of genetic variation when a new population is established in a very small area. This is, for example, if I were to place a small group of lizards on a new island, they are founding a new population, and the traits and their genetic diversity only comes from those founding organisms. Biologists got a chance to study this when a hurricane wiped out all these lizard species in the Bahamas, and they were able to look at how a specific species and the gene pool looked at different islands. This is the example that we talked about, right? The idea that the anolis or the anole lizards populate different areas, and eventually they became different species. They were all one species at one point. We see that essentially they were able to select a couple different organisms, place them on an isolated island, and watch them breed, and look at their genetic makeup. They found that basically all of these lizards eventually adapted to their environment, but retained characteristics from the founders. An example of a human founder effect is an Amish community founded by some colonists. They contain genes for polydactyly, which is extra toes and fingers. The Amish population has increased in size, but has remained really genetically isolated. They don't reproduce with anyone outside of the population. Therefore, polydactyly is much more frequent in Amish communities because they started with a very non-diverse gene pool. They only bred with themselves, so there's no introduction of new variations. And so we see them passing on some pretty negative, not always harmful mutations, but some interesting mutations. The impact of non-random mating. So this is going to be artificial selection. Non-random mating can also change allelic frequency. Basically, if we think back to our frogs, and if we suppose that 4 frogs migrated from some distance to the main pond, it is likely that the 4 frogs will mate with one another, but not mate with that larger group of species. Non-random mating implies a choice of mates that results in certain genes staying within a group. Darwin wrote about this idea of sexual selection. There's this sexual struggle. One is that between individuals of the same sex, generally the males, they are in competition with each other. The females remain passive, whilst in the other, the struggle is likewise between the individuals of the same sex in order to excite or charm those of the opposite sex. And we see that it's not just random sexual mating most of the time. If we're talking about evolution, there's a lot more that goes into why we choose the mates that we choose. And so these are some things that can impact the genetic diversity. Sexual selection of mates affects allele frequency. There are certain traits that we see in our counterparts that we are attracted to and choose to mate with them because of those traits. This is going to shape the genetics that are passed on and therefore shape the allelic frequencies within the population. An example is a peacock. The more fantastic the spread of the feathers is, the more likely they will get to mate and pass on their genes. Therefore, the genetic pool for having these very impressive feathers is going to increase, and so therefore impacts the allelic frequency. Female birds of many species choose males, and so besides our peacock, we also see that in other species the males are supposed to be like showier or more colorful. When we think about, especially in birds like the cardinal is a red bird, the female is like a brown bird. These males have a selective advantage. They are more susceptible to predators, but the showier they are, the more likely they are to pass on their genetics. Again, this impacts the genetic makeup of the whole population pool. Females are drab because they blend in. They need to be able to survive and take care of offspring. This shows that sexual selection can lead to sexual dimorphism when there is a big difference between males and females. Sexual dimorphism means that there are physical characteristics that are different between the males and the females. We're going to pause here. We're going to talk about Hardy-Weinberg equilibrium next. This is an equation that you need to know and need to be able to use to calculate percentages of allelic frequencies, and we will get into this in class.